All right, it is 12:02, so we're going to go on and get started with the April Research Town Hall. Appreciate everyone joining us today. We do have quite a bit of a great agenda with different presenters from the Center for Integrative Environmental Health Science. And so we're going to start it off with an update from Dr. Klein.
Okay, well, good afternoon, everybody. As in most of these sorts of sessions since February 7th, have the task of talking to you about the status of grant submissions as well as grant funding in light of the executive orders from the administration. Let me tell you first the approximate number of grants that have been terminated, and that is approximately $7.5 million in grants have been terminated. And this was actually 9 million at one point, but we got one grant back that was reactivated, which was a pleasant surprise. Most of these are related to supporting students in terms of the numbers. The largest number of grants in a category are supporting graduate students in programs that were developed to address the problem of underrepresented minorities in science. Those are canceled, and so we are scrambling to find ways to support those students. There are other ways in which the flow of funding have been interrupted, and I want you to be aware of them. Of course, the first one is termination of grants. I want to reach out to everybody on this call and tell me that, please let you know that if a grant is terminated, we actually hear in sponsored programs sometimes and frequently after the investigator does. So if you have had a grant terminated, please let us know. Forward the email describing the termination so that we can add it to our running total. There really has not been a huge ramp up in the number of grants, but each one that's canceled is painful. And it's important for us to be aware so that we can position ourselves to deal with it. That said, the other way that the flow of funds can or has been interrupted or slowed, first is of course the executive order dealing with indirect costs. That's currently blocked by a permanent injunction, but it is possible that the judge that's hearing the appeal could get relief to the administration and allow that to go forward while they are considering the case. We don't think that'll happen. It's rare that it happens, but I just want everybody full disclosure and awareness to be aware of that. And so that's the second thing, is that the indirect cost dollars could fall abruptly, not likely, but could. And the last one is that there's a whole series of roadblocks that are being put up asking us to essentially itemize invoices on the grants that we have received and to be specific in our invoices. This is something that's never happened before. So far, Sponsored Programs has dealt with this really well, and the money has continued to flow. Big shout out and tremendous gratitude to Irvis and to Bryn, who are working weekends and evenings to make sure that, and their whole team, to make sure that this continues to flow. The exception to that is the Substance Abuse and Mental Health Services Administration. Grants from there, they're a little sticky on the invoicing process, but we're figuring that out, and I don't anticipate any interruption in funds. So that's as much bad news as I can give you. The good news, and I think it's really important for everyone to understand that our work is continuing and that the good news is still there. So we've had a couple R01s that got notices of award this last week. We had an S10 that got a notice of award this week for a substantial amount of money. Just a teaser here. Next month on this town hall, we should be able to share with you very good news about a large grant that we expect to get the notice of award in the coming days. So with that, I'll throw it back over to Lee, and I think our first speaker is actually not Dean Bumpus, but I think it is Dr. States, who's going to tell us a bit about research at the medical school, and then also followed up with some news about the CIEHS. Anything else to add, Lee?
No, the only question I have for you, when people are reaching out to our office, if they are getting terminations or communication in general, do you want them to send that to the OSPA service account, or where should they direct their communication?
It should go to the OSPA service account, which is being checked multiple times daily under the current management. There does not appear to be a backlog. So that's a reliable place. But please go ahead and copy me on those so that I'm aware of them as well.
All right, thanks. I'll put that in the chat here in a minute. And like Dr. Klein said, and I said earlier, we are here to, our focus today is to talk about the great things going on in the CIEHS. And so I'll turn it over to Dr. States.
Okay, well, thank you for the invitation. So from the Dean, I guess, and perhaps Dr. Klein can comment on it as well. Progress is being made on the floor plans for the new building on the HSC campus. School of Medicine is wrapping up its progress on the 2023-2025 strategic plan and organizing to start the next strategic plan. Dr. Klein commented on the monitoring of NIH grant situation. We do that as well. Perhaps Dr. Klein might want to comment on the latest White House proposal for reorganizing the NIH. It's even more severe than the congressional proposal. They are going to eliminate, or they're proposing to eliminate institutes focusing on nursing research, alternative medicine, minority health, and global health research. And they're consolidating 15 of the remaining 20 into 5 new institutes dealing with quote unquote body systems, neuroscience, general medical science, disability, and behavioral health. Cancer, aging, and infectious diseases institutes, surprisingly, are all being left alone in the White House proposal. So other news from School of Medicine is we now have an Associate Dean for Clinical Research. That's Dr. Jai-Pen Huang. And he also serves as the Chief Scientific Officer for University of Louisville Health, the hospital. And he's Deputy Director of the CTRD/LCTRC. So he is leveraging all those positions to really invigorate clinical research and clinical trials here at UofL. So that's the news from the School of Medicine. I don't know if Dr. Klein wants to comment on any of those.
Yeah, just to say that we're keeping an eye on the proposals to reorganize the NIH. We've communicated with our lobbying group in Washington. What we feel about the reorganization is that some of it may be a good idea, some of it may not be a good idea. I think we're more focused on the White House's budget, which proposes a 44% cut in the NIH, and that's something that there's really not a lot in terms of intervening that we can do, but in terms of monitoring and communicating with the faculty and with staff, that's really the role for this office. I think the other thing that you mentioned is really worth noting is Jai-Peng's position puts him ideally situated to promote clinical and translational research across University Hospital and the other 8 UofL Health hospitals. And we're already seeing substantial progress towards constructing a a data warehouse off of the EMRs. And then also another kudos to Ma Yinkong and Jaipeng and others who submitted an NIGMS grant just this week, I guess last week, kind of running together, but to fund outreach of the data warehouse to not only UofL Health, but also to Owensboro Health, and eventually to Baptist Health, all the way to Paducah. So there's some really positive things going on. It's easy to lose sight of that. You know, we're all very stressed, and there's sort of this, you know, almost daily, you know, shock to our systems. But it's important to stay focused on the things we're good at and that we're succeeding at, and those are a couple of them. Back to you, Chris.
Okay, so I'll share my screen and start the CIEHS presentations.
So Chris, before you do that, we do
Yeah.
have one person in the chat is asking, what did you say about the administration at NIH and what the NIA is talking? What will happen to that? What's the rumors?
The NIA, well, aging is being left alone.
Okay.
There's an article in Science. This. So what we know of the details are enumerated in this article.
Okay.
Okay.
And just let me point out that there's a long road between now and a budget in its final form, and that Congress historically has protected the NIH. We will see if they're willing to do that again this year. But you know, there's always going to be extreme proposals, and if we react and get anxious to each one, which is only human, the best thing we can do is then turn around and go back to work and wait for the process to work itself out, in my opinion.
Definitely. Just be aware of what's out there and write your congressman. Okay, so the Center for Integrative Environmental Health Sciences is a National Institute of Environmental Health Sciences P30 Core Center and has been supported by this grant for the past 5 years. And hopefully the renewal will pass the approval of DOGE. And I have the privilege of directing the center. So it's one of only 26 environmental health sciences core centers, and you can see them all arrayed here on this map of the US. So we're in a relatively elite club. And what are the purposes? So it's basically an infrastructure support center to foster and support environmental health research. We also promote community engagement and education, promote research translation, and primarily also stimulate new collaborations and particularly new NIEHS research grants. We also recruit and mentor junior faculty and recruit other more senior investigators to environmental health science, and we support facility cores. So our objective is to build a framework to integrate the interactions between pollutant exposures and lifestyle factors and to assess their impact on human health and disease, also as modified by life stage, genetics, and sex, hence the integrative nature. So our aims are to promote interdisciplinary collaborative research in current and new areas of environmental health research, to translate basic research findings, and we use and work within the research translational framework shown here on the right from the NIEHS. And we recruit new and established investigators to environmental health research and promote the community engagement and community-based research. And you'll be hearing from some of those after I finish. So we are truly interdisciplinary. There are 66 faculty members representing 19 departments and 7 schools across the university. Yes, the majority are from the School of Medicine, but we have members from law, nursing, engineering, public health, the Kent School of Social Work, and the College of Arts and Sciences. We have a pilot project program And these are to acquire data to support R01 applications, preferably to NIEHS. We have several categories: interdisciplinary, where we look for collaboration between MPIs from different disciplines. New directions, so if a PI wants to change and go into a new area of environmental health research or come into environmental health research, that's a good category. We have a career development category where we partner with departments in recruiting new tenure track assistant professors and give them a pilot project grant. We have community-based research grants and emerging issues, and I'll talk about one of those in a bit. So far, over the past 5 years, we've awarded 23 grants for nearly $1 million total. So those grants all require preliminary data, but we also have a core usage subsidy program, which is equivalent to the voucher system from the EVPRI's office. These provide funds for collecting data from facility cores to support applications or to augment existing NIEHS-funded projects, so to expand the scope. The facility cores of the CIEHS provide guidance on experimental design, e.g. for clinical trials, in addition to providing guidance in omics services and helping to support omics services and data analysis in the bioinformatics and biostatistics core. The amounts range from $1,000 to $10,000, and these are paid directly to the core. So far, we've given 45 awards totaling a little over $250,000. So our region of interest is Western Kentucky, including Louisville. And this is our theme is an urban exposure and rural environment in most of Western Kentucky. And within Louisville, we actually have rural exposures in an urban environment at the backside of the track. So here in Western Kentucky, very high cancer rates, high cancer risk in Hawesville, Henderson. There's a noted PFAS problem there. We'll talk about that. Calvert City has super high cancer risks, and we'll talk about a project going on there. And in Trigg County, we helped the community with a hemp processing plant and getting that moved out. So the air quality in Trigg County was negatively impacted by a hemp processing plant, which polluted the area so bad, some of the residents said they couldn't see to drive on the road. You can see it's a very rural area, and we had members go down and provide air monitoring and other assistance in explaining some of the toxic potential of these things and hearing the community feedback and their concerns. So I mentioned Henderson, Kentucky, and the PFAS exposure problem there. So there was a story on NPR, the local NPR station, by Ryan Van Velzer, who was the environmental reporter at the time, and keyed the community engagement core into that, and Dr. Jamie Young and ESI in the center organized with the local high school science club to do PFAS monitoring in the groundwater and in the soil. We also supported the presentation of a feature film, Burned, Protecting the Protectors, which is a film, a documentary about PFAS in firefighter gear, and Center member Banrita Walang, who you'll hear from shortly, was a featured panelist, and this event was at the Speed Art Museum. Currently, we have a team that's working in Calvert City, which is a National Priority List Superfund site. And you can see up here in the corners are the Tennessee River. And all along the river is this industrial complex with a number of industries, most notably Westlake PVC, which is the issue at hand. And the town, Calvert City, is a small town, 2,600, and you can see all the agricultural fields all around. And So what's going on is that hemangiosarcoma, which is a very rare liver cancer associated with vinyl chloride exposure. First linkage was made here in Louisville by Dr. Tamburo in his studies in the Rubbertown area with PVC plants there. And so Westlake PVC has a history of vinyl chloride and diethylene chloride leaks. And a leak over the winter holidays prompted the community to actually contact the center. And so our community engagement corps organized some community meetings where we heard from the community. We learned that there were 5 hemangiosarcoma diagnoses that have been documented over the years, including one recently, which is actually non-occupational, which is very concerning. So our community engagement corps is collecting urine, blood, and wastewater for analysis for biomarkers, and they've applied to NIEHS for a rapid response R21 to analyze those samples for biomarkers of exposure, and will return the data to the community. So with that said, our featured presentations will feature senior investigator, Dr. Carolyn Kling, and she'll talk about her exciting work in epitranscriptomic changes in mouse liver induced with high-fat diet and PCB exposures. And as I mentioned, Dr. Banrita Walang will discuss her work on food contaminants and chronic diseases. And then finally, Dr. Becky Antle will wrap it up with a community-based participatory research project that is funded by a pilot grant. So I'll answer any questions to my presentation.
Thanks, Chris. I think we can move on. Very timely subject here. Carolyn, if you wouldn't mind sharing your slides. Carolyn, we cannot hear you.
Carrie, you're still muted.
No, it doesn't look like she's muted. Why don't we have you maybe log off and back on, and we could go on and move to Banrita's presentation. Sure. And if you need to, Dr. Kling, you could send your slides to Cecilia Pareto, and I'll put her email in here, and we can maybe handle that, and you'll be able to talk if we have still problems.
I do have the slides already, Lee.
Okay. It's hard to tell if she's on. All right,
Are we going to move to Banrita?
Yeah, let's do that.
Hi, can everybody see and hear me?
Yes.
Yes.
Perfect. I'm just going to go ahead and share my screen, and please let me know if you see my screen.
We do, but it's in the workbook.
Okay, I will fix that. All right, how about now?
Great.
Perfect.
All right. Okay, well, thank you so much for this opportunity to present at a research town hall. I've never presented at a town hall before, so this is exciting for me. My name is Benrita Watlang, and I'm assistant professor in the Department of Medicine and have a joint faculty appointment at pharmacology and toxicology here at the University of Louisville. And we'll talk today about food contaminants. So what are food contaminants? So these are generally undesired materials that are present in our food, and they could be introduced into our food right from the agricultural or farming processes all the way to when that food is packaged, put on a shelf, and then put on a plate for us to eat. And these undesirable materials are generally pose a harm to the health of the consumer that's consuming the food in particular, and to public health in general, and therefore they are known as contaminants. And so food contaminants are broadly classified into three different types. So the first one are physical, so anything tangible that could be seen in food like metal, dirt, plastic, and usually attributed to carelessness in the food preparation process. There could be biological contaminants, also known as microbial contamination, like bacteria, fungi, viruses, and mold, and so on. And these are usually the main culprits of food poisoning in our day-to-day lives. And then the last category are chemical contaminants. And these are any kind of chemicals in food, like generally recognized as safe, could be additives, preservatives, and importantly, it could also be industrial chemicals, which is part of what we study, also known as toxicants. And so these toxicants are, they were not intended to be put in the food, and they just happen to be there by accident or happenstance. And so they're often viewed as inadvertent contaminants, and they include a list of chemicals, and just name a few here, industrial chemicals like PFAS and PCBs, dioxins, pesticides and fungicides and herbicides, as well as metals, heavy metals, and so on. And so part of the research that we do here in my lab is we focus a whole lot on these industrial chemicals, such as PCBs, dioxins, and pesticides. So what about chronic diseases? So in terms of chronic diseases, we focus a lot more on metabolic disease. And when one thinks of metabolic diseases, one think about the metabolic syndrome, which is this constellation of pathological conditions like obesity, high blood glucose levels, dyslipidemia, hypertension. And these are often risk factors for conditions like diabetes, obesity, as well as steatotic liver disease. And when we think about steatotic liver disease, generally people have often associated that with this metabolic dysfunction associated steatotic liver disease, which was formerly known as non-alcoholic fatty liver disease. It has a global prevalence of about 25%, so pretty considerable, and it also has a similar prevalence in the United States. And often, historically, metabolic dysfunction, steatotic liver disease, has often been blamed on hypercaloric diets, genetic predisposition, as well as sedentary lifestyles, like lack of exercise, physical activity, and so on. And now there is another subset of this metabolic dysfunction associated steatotic liver disease that's linked to chemical exposure, and is termed as toxicant-associated steatotic liver disease. And this was back in the day characterized in chemical workers, occupational and industrial workers for obvious reasons. But over the last decade, there has been a lot more studies that have looked at toxicant-associated steatotic liver disease in terms of chronic day-to-day exposures to environmental chemicals in the general population. So despite the etiology and the steatotic liver disease the development, the pathogenesis of the disease itself is somewhat similar. So you start off with having a normal liver that could then have steatosis or fatty depositions in the liver cells, and this could be accompanied by inflammation, and also known as steatohepatitis, and if left untreated, could progress to fibrosis, tissue scarring, and cirrhosis, and eventually to hepatocellular carcinoma, which is a liver cancer type. So in our studies, when we look at these toxicant exposures and their effects on metabolic diseases, we often have to think about organs in terms of their interactions, and also about other factors, because nothing is as simple as it looks like. So back in the day, there was this conventional thinking that chronic diseases like diabetes and obesity, and even steatotic liver disease, is often brought upon by this imbalance in energy intake and energy expenditure, so energy output. So we eat too much and we do too little to spend that energy, and hence we're getting obese and so on. But now we know that it's much more complicated than that. So there's a number of factors that could contribute to these chronic conditions. It could be what we eat, what we drink, and how we live, right? It could affect multiple organs like the liver, the pancreas, and the gut. And you add that to that, these environmental factors such as chemicals, environmental stressors, and lack to clinical care even, and that could all contribute to these chronic conditions like diabetes, obesity, and metabolic associated steatotic liver disease. And so with that in mind, one of the approaches that we adopt in our research program is to look at these mixtures of these toxicants from this broad perspective and look at these different pathological conditions that it could affect. And so we also keep in mind that we know that biological sex does play a role in dictating our chemical exposures and how our body responds to chemical exposures. And so we also have a focus on sex differences as well, and we look at how these toxicants impact multiple organs and how it could disrupt those organ-organ interactions to produce or result in metabolic dysfunction. And so this is a study, a snapshot of one of the studies that we do in our group. And so the study was focused on looking at what are these sex-dependent effects of food contaminants in the context of of steatotic liver disease, as well as trying to identify potential mechanisms such as metabolic and endocrine disruption. And so in this particular study, we use mice as our experimental model, and oftentimes we take the studies or the principles and concepts that we learn in the lab and translate that to human exposure studies. And so in the study, we had male and female mice that were early adults, if you will, 6 to 8 weeks old, and they were fed either a controlled diet, which is a low-fat, low-carb diet, or a Western diet, which is equivalent to a high-carb, high carbohydrate, and high-fat diet that oftentimes is, represents what we eat in Western countries. And so we had a 12-week exposure study where we fed these mice these diets, and then we exposed them to these mixtures of toxicants. And so the toxicants that we used for this particular study was chlordane, which is a legacy organochlorine pesticide, and a PCB126, which is a dioxin-like PCB, industrial chemical that was produced back in the day. And so the doses that we utilized or were designed for the study were based on what we measure nowadays in our general population. So they were extrapolated from levels that were reported in the CDC chemical exposure database. And then we perform a bunch of systems toxicology assessment to kind of look at these, effects, overall effects and metabolic disruption. And so just a snapshot of what we found. So we found good and bad things. So in general, this is just a little a slide just showing you one of the major findings. So we saw that when we fed these male mice this Western diet over that 12-week period, they did develop this lipid accumulation in the liver, as you could see here by these histological depictions. But when you expose them to that chemical as well, we didn't see this augmentation in fatty acid, or lipid deposition in the liver. So, which is a good thing. So we didn't see really any harmful effects, so that's great. But unfortunately, what we saw was that when we look at our female mice, they didn't have as much fat accumulation in the liver with this Western diet in general. But when we exposed them to both the diet and this chemical mixture, they did tend to have a higher frequency of lipid droplets in the liver, suggesting perhaps that, there are these biological sex chemical diet interactions at play here that could dictate the susceptibility to metabolic disruptions by these chemical exposures. And so another thing I wanted to share with you, again, this was another study that was funded by the CIHS CUPS Award. So we also wanted to understand what these chemicals do to our gut microbiome, because we're trying to look at that holistic picture of these phenotypic effects. And so this is another study that we did where we just look at a mixture of these PCBs, and it was an acute study, one-time exposure for 2 weeks in these mice. And what we saw was that there was these differences in microbial composition that resulted from exposure, and it was also driven by biological sex. But what was interesting to us was that we saw that there was this enrichment in this bacteria known as Dehalobacterium in our male exposed mice. And this bacteria, we don't generally see it in our normal experimental mouse model, but with exposure, we kind of saw that enrichment, and it suggests to us that perhaps these differences in microbial signatures that, we see with chemical exposure could be exploited perhaps as a non-invasive method to analyze or assess chemical body burdens in humans. And so with that, I would like to end this 10-minute talk by having some conclusions of, the status quo of what we're at in terms of food contaminants and chronic diseases. So what are we as a community talking about? What are those conversations that we're having as a society? And there's a lot going on, but I'd like to point out 3 things here. So the first thing is that we should acknowledge people's fears, people's doubts, and their questions about food safety. But it's also important to have those really important legally binding agreements, if you will, between regulatory agencies as well as food companies that perhaps are not just performative politics, but could actually result in actionable change. And lastly, as the father of toxicology, famous words, the dose define the poison. So I think that's important to remember that studies such as the ones I'm showing you right now are important to give this holistic picture about, food safety and what food contaminants can do to our health, as well as the good, the bad, and the in-between. And with that, I would like to end this little talk by acknowledging everybody here at UofL, students, staff, and faculty that's always been supportive and contribute so much to my research program. And I would be happy to take any questions.
Thank you. That was very interesting and exciting. Does anyone have any questions before we move on? All right, I'll circle back to see if Dr. Kling is able to join now. And if not, we'll go on and move to Dr. Becky Antle. Anybody know? Let's see if
Dr. Kling's mic is on.
Are you able to speak, Dr. Kling?
Okay, I'm switching over to my laptop. Usually I use my, or my desktop. Usually I use my laptop for presentations, but we can try it this way. Does everybody, oh, I guess I have to do this slide share thing. Okay, so back over here.
Or Cecilia could put them up.
We'll see who can get it faster.
Are you seeing my
There
screen
we
now?
go. Yep.
Yes,
Looks
we
good.
can see it.
Okay, great. Then I'm delighted to share with you some of the work that we've been doing in my lab looking at epitranscriptome changes in mouse liver with high-fat dye and PCB polychlorinated biphenyl exposures, both in vivo and in vitro, and I have nothing to disclose. So, what is epitranscriptomics? What it means is the chemical modifications of all transcribed RNAs. These are tRNAs, ribosomal RNAs, non-coding RNAs, and mRNAs. These chemical modifications can occur on each of the 4 nucleosides as indicated here. The most common modification in all transcribed RNA is N6-methyladenosine. And that can occur throughout the mRNA transcript from the 5' UTR through the coding region and into the 3' UTR. But it's still a very rare event from an average of 3 to 5 M6A sites per transcript. However, because of its importance in RNA biology, what you can see is increasing interest in terms of the number of publications per year in which investigators are examining the impact of M6A. Now, M6A is applied, the methyl group is applied to adenosine by a metal 3 transferase complex at specific sequences called Dratch motifs within transcribed mRNA. Now, the importance, this is a reversible mark, and so it can be erased by ALK-BH5 and FTO, which are demethylases. Now, the importance of M6A in RNA biology is endowed by 18 different reader proteins, which recognize that M6A mark and therefore regulate splicing, transcript stability, intracellular localization of the transcript, and translation as well. So what's the importance in terms of disease relevance? Van Rita just told us that MASL, metabolic dysfunction associated steatotic liver disease, has a prevalence of about 24% in the US population. And that's been increasing due to the obesity epidemic, as well as our exposure to metabolism disrupting chemicals. And these are environmental chemicals to which we are all exposed in terms of water and food and air. And Dr. Matt Cave has done pioneering work in which he investigated the association of MASL with exposure to polychlorinated biphenyls in an exposed population. Now, these weren't people that were only exposed occupationally, but were also just in the same town in which PCBs were manufactured. Now, PCBs are persistent organic pollutants, so they are legacy compounds, and we all, or nearly all of us, have body burdens of PCBs within our adipose tissue. Dr. Cave and Ben Rita was part of his group as well, established a mouse model in which male C57 black 6 mice were exposed to a high-fat diet for 2 weeks and then given a single oral exposure to different PCBs. Aerochlor 1260 is a mixture of PCBs, and PCB 126 is a single congener. Either these two individual components together or in combination. And this resulted in a compromised liver with MASL that can progress to MASH, metabolic associated steatotic hepatitis, or as called by Dr. Cave, toxicant associated steatohepatitis. Now, I was interested in working with Dr. Cave's group to determine whether changes in transcriptomic modifications, particularly M6A, was involved in the progression of a disease from MASL to MASH. And so Kelly Peel in my lab performed M6A RNA immunoprecipitation followed by Illumina RNA sequencing to identify, with the help of Kalina Andreeva and Eric Rushka, individual changes in genes on the right with the different Aerochlor, with the different PCB exposures. Here's Aerochlor 1260, PCB 126, and the combination of Aerochlor plus 12CB, PCB 126. And this was the first time anyone had ever seen that these pollutants could alter the M6A epitranscriptome in RNA. And so we found that M6A modifications play a role in PCB-induced reprogramming of hepatic gene expression. And we did this by comparing also the microRNA-ome and the mRNA transcriptome of these same livers. And we identified with Metacore pathway analysis changes in lipoprotein metabolism, which is a hallmark in MASL and MASH, and also changes in ApoB. We saw an increase in M6A in the ApoB transcript that was associated with less ApoB transcript abundance. So we hypothesized that M6A destabilizes the ApoB transcript or inhibits its translation. Thanks to the support of the CIEHS P30 voucher program it has allowed us to continue on this work from the first NIH R21 grant that we had and to collect preliminary data. And so the first voucher paid for global modification analysis of the entire epitranscriptome, so that would be RNA, tRNAs, as well as mRNAs in this mouse model of TASH that we just looked at. And this was performed by Cream, and what we've identified were different changes in the global epitranscriptome, including changes in tRNAs and specifically. We then followed up with a different model in which mice were fed a normal diet rather than a high-fat diet. And after 2 weeks, were given a single oral gavage of Aerochlor 1260, that mixture of PCBs, and they developed MASH or TASH. And we performed global epitranscriptomic analysis, and we identified, interestingly enough, changes specifically that are hallmarks of selenocysteine tRNA. And so that was followed up with another medium voucher grant that allowed us to look at the metalome, in other words, the metal composition of the liver, as well as using an unbiased proteome approach with Mike Merchant and the metal analysis was performed by Dr. Lukai. And we identified changes in selenoproteins in the liver of these PCB-exposed mice with MASLD. And as you can see, we've published this work with the support of the P30 grant. More recently, Dr. Cave was awarded a large voucher that allowed us to do IsoSeq, which is long-read RNA sequencing, allowing us to identify alternative splicing events in the livers of these mice. And the work was analyzed by Dr. Julia Charkar in KY Inbury. And here we've put together a control versus the Aerochlor 1260 exposed liver samples, and we identified an increase in splicing events in the liver of the Aerochlor exposed mice with a high NAS, which stands for Natural Activity Score, means more liver injury in those particular mice. Well, you know that these CIEHS core funding of voucher program is to enable investigators to collect preliminary data for NIH grant proposals. And indeed, this data has been used in this grant application that I submitted to NIEHS, and it got a score. And what I wanted to do was to identify a tractable system in which we could modify specific M6A at individual sites within the ApoB transcript in mouse hepatocyte cell lines. We can't do that in livers, but we could do it in mouse hepatocyte cell lines. And of course, the reviewers came back and said, well, you have to show us that you can do it before we're going to give you the money to do it. And so we obtained another CIEHS medium voucher. In this case, we used AML mouse hepatocyte cells and exposed them to palmitic acid. And this was this is a BSA conjugate, and this is mimicking the high-fat induced steatosis that we observed in the mice in vivo. And the cells were treated with either DMSO or the combination of the Aerochlor 1260 plus PCB 126, because we'd seen the most epitranscriptomic changes there. We did 5 biological replicates per group. And this time we performed direct Oxford nanopore technology RNA sequencing. And so how this works is that now this is direct. So instead of going from RNA to cDNA, the RNA is directly read. So the RNA molecules are translocated through this protein pore, and then which is embedded in a membrane. And this causes disruptions in the electrical current that is read on the MinION. So this is Kelly Peel performing the library prep and then loading the nanopore device, and then we get a readout. This readout of this electrical readout is read by machine learning, namely a program called M6A Net, which has been utilized by Dr. Eric Rushka and Dr. Belinda Petrie, who's a postdoc on this project. And what this M6A Net gives us is the probability of an M6A modification at a specific site within an RNA transcript, and the modification ratio, for example, between treatments. So we have AML12 cells treated with DMSO versus PCB exposed AML12 cells. We have direct RNA sequencing, and in parallel, we did Lumina short read RNA sequencing for integrative analysis. And what do we know thus far? Thus far, we have seen transcripts that are increased with M6A abundance or decreased with M6A abundance. And so the big question is ApoB there? And thankfully, it was at a p-value of less than 0.01. So M6A is altered in ApoB in AML12 mouse hepatocytes, similar to what we had seen in the mouse liver. So then, of course, you say, well, is it the same site? Because if you're a KG NIH R01 reviewer, you'd want to know that information, right? And so here, Belinda has aligned ApoB genome view. And so ApoB has 29 different exons in 4 different transcripts or splice variants. And here are the data, the M6A sites from our M6 RIP sequencing. That's going to be broader than the single base pair resolution of the direct nanopore RNA sequencing method. But what we can see, and we can blow this up, is that some of these M6A modifications that we identified in the AML12 mouse hepatocyte cells are the same as what we saw in the high-fat diet fed mice exposed to the same combination of Aerochlor 1260 plus PCB 126. And so that data was included in the A1 of this R01 that was submitted last November. It was supposed to be reviewed by EDD on March 6th. It now will be reviewed on the 29th. So I hope you will all keep your fingers crossed for a fundable score on that R01. And that's all I have to say about it. We have published 6 papers, thanks to, in large part, to the core support from the CIEHS program and also to funding from the Jewish Heritage Fund for Excellence. I'd be happy to answer any questions that you might have.
Thank you, Dr. Kling. And also thank you for both of the presenters so far for keeping to their time that they have been allotted, because that gives us sufficient time for Dr. Becky Antold to share her research. And so I'm going to turn it over to her.
Thank you. All right, I'm going to be talking today about another pilot project funded by the CIEHS. This is a community-based participatory research project to understand environmental health risks and needs for special populations. Specifically, we were focused on newcomers from African and Latinx countries who live in West and South Louisville. We conducted a comprehensive needs assessment using a mixed methods approach, and then using the data from that needs assessment, developed educational workshops and other resources and materials to address some of the needs that were identified. In addition to myself, I've been working with Dr. Luz Huntington-Moscos, who is also the head of the Community Engagement Core for CIEHS, and she has been really instrumental in the implementation of this pilot grant, and our project leader is Tiana Stewart at the Kent School. I am on faculty in the Kent School of Social Work and Family Science, and also the director of a research center called the Center for Family and Community Wellbeing. I also serve as the evaluator for CIEHS. So in terms of understanding our target population for this needs assessment project, the newcomers that we are focused on predominantly live in West and South Louisville. When folks arrive here from those countries of origin that we mentioned, from both African and Latinx countries, they are predominantly placed in apartment complexes and rental housing in West and South Louisville. We worked with 2 partner organizations, See Forward Ministries, which serves the African community, and Adelante, which serves the Latinx community. They live in very similar areas in the Louisville zip codes. And what we know about zip code is that zip code is still the most potent predictor of an individual's health and well-being. And that is certainly something that we have seen in these areas and has been an area of study through a number of important projects that you're likely aware of with the university. So we know in terms of West and South Louisville, there is a high concentration of industrial facilities, limited groceries, the proximity to the airport creates noise and emissions from planes, and we see higher cancer rates and asthma rates and lower life expectancy. What we don't know is how does that interact with specific features of these newcomer populations, and that was really what we were focused on with this particular needs assessment and educational intervention development project. We did have some pilot data that helped drive this effort, including a global needs assessment study that we had conducted in partnership with See Forward back in 2023 that was completed by 537 members of the African community and identified risk factors such as overcrowded housing, poverty, and unemployment. And then a focus group with South Louisville neighborhood place, where again, issues such as noise, water, land, and air pollution were identified as key concerns of community members. From a methodological standpoint, this was a mixed methods needs assessment study. Initially, on the qualitative side, we conducted focus groups. On the quantitative side, we administered surveys. And this again was with 2 different target populations. So with See Forward, we conducted 8 focus groups that were stratified by newcomer status, so basically length of time in the country, age, life stage, and gender. And we also distributed the survey to over 90 community members, and the survey actually built upon the results of the focus group so that we could gather more in-depth data from what we learned in those preliminary focus groups. We also worked with Adelante to assess the Latinx community. where we did 2 focus groups, had multiple community advisory board meetings, and the survey is still in progress. The questions for both the focus groups and surveys focused on environmental health exposures, housing and neighborhood issues, health and healthcare utilization, and other barriers. All of the materials had to be translated. For C Forward, the materials were translated into 5 different languages. For Adelante, the materials were translated into Spanish, and we also offered live interpreters at all of the data collection events. The most critical piece of the methodology for this research, though, is the community-based participatory action research nature of our work. And in that, we mean that we formed these community advisory boards. That was very easy with C Forward because they have existing ambassadors who represent the different neighborhoods that C Forward serves. And with Adelante, we formed a community advisory board, also pulling in from 2 other agencies, Backside and Neighborhood Place, to serve the Latinx community. And the community advisory board served several critical functions in this research, not only sharing their lived experience to help guide our understanding of environmental health issues facing the population, they also provided direct consultation and input into the research and the formulation of the questions for the focus groups, and the vetting of the questions for the survey research, and the interpretation of the data prior to presentation to the community, and in the prioritization of issues to be addressed through the educational interventions. They were also critically important in supporting recruitment, facilitation, and data collection processes. So very briefly, from the focus group, some key environmental health and healthcare findings. On the environmental, and these are word clouds that we often generate from our qualitative research. On the environmental health side, we did see in multiple focus groups and the thematic analysis of over 120 pages of text that there were issues related to overcrowding, air conditioning and heating not working, neighborhood cleanliness and safety. These things had impacts such as skin disorders, sleep issues, hospitalizations due to various issues. And there were actually 2 tragedies in the community, one death due to carbon monoxide poisoning, and another where a child drowned in an unsecured pool. Much of this actually related back to a general lack of awareness and understanding of how to mitigate some of the risks that they were experiencing in their apartments and rental housing, and difficulties in advocating for improvements and repairs. Many of the folks that we were working with, specifically with C Forward, did not have experience in the type of housing and did not have basic awareness of how to mitigate some of those risks. In terms of the specific issues people were experiencing, there were a lot of issues with insects, general cleanliness, mold, and then, as I said, fairly low levels of understanding of how to navigate various housing issues. We did see that the number of housing problems people reported was significantly different based on the length of time they had been in the US. So the less time people had been here, the more issues they were experiencing, suggesting that as people were here longer and learned more about managing housing and navigating various systems, the safety of their housing situations improved. They did think there was a perception that this was having an impact on their health, and they reported a variety of symptoms, and we did see a significant positive correlation between the number of housing issues and the number of health issues reported. There were also a number of barriers to accessing medical care. 30% of respondents indicated they don't obtain necessary medical care because of cost, and many rely on emergency room services. And even then, if there are language barriers, they don't access even emergency services. This is just more on, again, healthcare and healthcare utilization. There's also a key theme that came up around mental health issues. And this is actually one of my substantive areas of expertise and focus over the years. And so we've seen a high rate of reporting of stress, anxiety, and depression, and then stigma around help seeking. And even, again, one community member who tragically died by suicide as a result of lack of services. So as a result of what we've learned from these surveys and focus groups, our next steps have been to work with C Forward to develop some programming around housing and neighborhood safety. This is a workshop that that not only have we developed with them and will be offering as a part of this pilot grant that will finish up in June, but they'll be able to offer in an ongoing way to all newcomers upon their arrivals to help them understand how to better mitigate some of the risks that they're experiencing in their housing and neighborhoods. We also have developed a workshop around advocacy and how to navigate things like landlord issues and community issues. And then lastly, because of the number of barriers people were experiencing related to medical care, we also have offered already 2 clinic days where we're doing some basic health screenings. We did that actually last week and we saw about 300 people, and then we'll do several more of those before the end of the grant. And through those resource fairs and workshops, we're distributing a number of resources and for people's homes and health maintenance that they can use beyond the funding of the grant. We are finishing up our research with Adelante. We have seen similar themes there in terms of issues with air and water quality, drainage and sanitation, housing conditions. And again, a need for information and advocacy support and affordable solutions. So we will be offering workshops to Adelante in May and continuing to work with them as long as we have this funding through June. And obviously our hope is to continue to pursue additional and larger funding sources to build upon this work so we can take more direct measures of some of the environmental risk factors that were identified through the focus groups and surveys, as well as more biological measures of health issues. We did identify through the clinic days some of the more prominent health issues that people were experiencing as we were doing direct testing, and we hope to be able to build on that through foundation funding as we move forward. And just to thank again the many members of our team at the center that assisted with both the data collection and the clinic days most recently. With that, I'll stop and see if there are any questions.
Thank you, Becky, and all of the presenters. Before we hop off, I just added to the chat a message about some very exciting grant writing tools that are going to be made available to campus at no cost to researchers or their departments and colleges. There is going to be a sneak peek of the course library at next Tuesday's LCTRC's Professional Development Corps virtual webinar. So if you're interested, and everyone should have received notification that this is available, but if you have not yet registered and need the information, you can contact Christy Treatment and she can provide you more information as well as the team's login. It looks like she just added the registration link. All right, thank you, everyone.
Lee,
yeah.
If I may, I want to close out with expression of gratitude to you and your team for seeing the Bouvier Group contract through, which will give NIH mentorship to a wide spectrum of people. And also a gratitude to your team. 2 enormous NSF grants were submitted. The so-called Innovation Engine grants, 2 of them from the university. It was a real heavy lift for you and your group, both of you. And we're all very grateful for everything that you do. Research development has more than shown how valuable it is in the last 2 years. So thank you very much.
You're welcome. And just to clarify, because I did see Johanna on here, we actually had 3. We also had one that Dan Popa collaborated with UK. So we had 2 subawards and one with UofL as the lead. And yes, we're very grateful that period of time is over.
And congratulations to Dan as well. I probably just sort of censored it because it was based at UK. But we'll make sure that we always mention those types of collaborations. Thanks, everyone, for attending.
Thank you, guys.